Good afternoon, everyone. We're going to give it just a few seconds for folks to load into the webinar and then we'll get started. Okay, good afternoon, everyone. My name is Jessica Regler. I'm with Blue Cross Blue Shield of Arizona. I'm excited to welcome you here today for our last Arizona Community Grand Rounds of 2023 on emerging infectious diseases. We've got a really great session planned today and some fantastic experts who I'll introduce to you in just a moment. First, I'd love to extend a thank you to our partner, the NARBA Institute, who's instrumental in making Arizona Community Grand Rounds possible. And specifically to Mary Jo Gregory, Dr. Mark Harrell, Carla Berg, and Jennifer Pierce for making all of this happen. Please make sure that you stay to the end of the session. At the conclusion, we'll share information about how you can obtain CEUs for your participation today. So now it's my pleasure to introduce our speaker panel with a brief summary of their very impressive bios. We'll hear from Dr. Lisa Villarroel. She's the Chief Medical Officer for Public Health at the Arizona Department of Health Services. Lisa has her bachelor's in biology at Princeton, her doctor of medicine from Northwestern, her master's in public health, a certificate in media and medicine from Harvard Medical School, and she's board certified in family medicine. Dr. Villarroel has served as medical director for public health emergency responses at the state level, including Ebola, Zika, opioids, vaping, and the COVID-19 crisis. And she's also been the medical director for the Division of Public Health Preparedness and the state's TB control officer. Next, I'd like to welcome Dr. David Engelthaler, who's the professor and director of TGen North, which is the infectious disease arm of the nonprofit Translational Genomics Research Institute. He's also the scientific director of the TGen North Clinical Laboratory, which was stood up in March of 2020 specifically to provide COVID-19 testing. Dave oversees a number of research groups working on detecting and understanding infectious diseases like COVID, tuberculosis, and valley fever, and provides genomic services to CDC, state, local, and tribal health departments and Arizona healthcare institutions. With those very brief bios, I would like to turn the floor over to these talented individuals. You can feel free to use the Q&A function to send questions our way. And at the end, we'll dive into any questions that we might have time for. So Lisa, Dave, please go ahead.
That's great. You can see my slides?
Yes.
Perfect. All right. So thanks so much, Jessica. And good afternoon, everyone. My name is Lisa Villarroel, Chief Medical Officer for Public Health at ADHS. I have been at the department for over 10 years now. So I was not there for H1N1, but I was for the emerging diseases of Ebola, Zika, chikungunya, West Nile outbreak, and COVID-19. Dr. Engelthaler and I have jointly created this presentation today on emerging infectious diseases through an Arizona-specific lens. So between the two of us, we're going to be providing the public health and the laboratory perspectives. The two of us have nothing to disclose. And I'm going to be starting us out on the definitions and current theories on emerging infectious diseases. Then I'll move into the history of some diseases that have emerged here in Arizona in recent decades. Then Dr. Engelthaler will get into some of the newer emerging diseases here, and then we'll end with some resources and take-home messages for clinicians. I think talks on this topic are always fun and they are ever-changing. So I hope you all have your lunch and can enjoy it while getting some CME at the same time. Okay, let's start with some basics. So, according to the National Institute of Allergy and Infectious Disease, where Dr. Anthony Fauci was the director previously, emerging infectious diseases are those that have newly appeared in a population or they've existed, but now are rapidly increasing in incidence or geographic range. So the classic examples here are HIV, SARS, Lyme disease, E. coli 157H7, West Nile, Zika, COVID, and so on. Then we can look at re-emerging infectious diseases, and those are the ones that reappear after they've already been on a significant decline. So we've seen them before, they've been experienced before, but now they're coming back in a more virulent form or in some kind of new epidemiologic setting. And the classic ones for those are listed here and include influenza and their pandemics of 1918, 57, 68, and 2009, and then malaria, TB, cholera, and pertussis. It is interesting how we're here at the end of 2023 talking about this because but emerging and re-emerging infectious diseases have been threatening humans since the Neolithic Revolution, when we were getting hunters and gatherers settling into villages to start domesticating animals and cultivating crops. And it was these beginnings of domestication that led to emerging zoonotic diseases, shown on the table on the right. These show some highlighted diseases from 430 BCE to 1981. And then this map picks up in 1980 and brings us through 2020. And it shows the global extent of newly emerging and re-emerging infectious diseases. It also includes, you see in black, the deliberately emerging diseases like anthrax, which is a category signifying a disease associated with intent to harm. The literature also uses now the term accidentally emerging infectious diseases. Those are diseases from pathogens created by humans that are released unintentionally, like epizootic vaccinia. I will take a moment and say that I see and respect all of these here, but for my portion, I'm kind of addressing all of these synonymously and simultaneously. I think the relevant part here to clinicians is the concept that there's a new disease to diagnose, treat, manage, and prevent, no matter where, when, or how, or the intent where it came from. So how do these new diseases emerge? Well, 70 to 75% of new pathogens causing infectious diseases do come from animals. They're often viruses that have been circulating in animals for decades or even centuries. And then at some point they jump or they spill over into people. For instance, HIV was introduced to humans from non-human primates in 1920s or maybe before, and then the pandemic followed in the 1960s through 1990s. There are other ways that we can get emerging and re-emerging diseases. It can come from de novo evolution of pathogens. Maybe those pathogens undergo some microbial adaptations, or there's other host, agent, and environmental situations. And speaking of those three factors, this is a version of a century-old diagram called the epidemiologic triangle. And we use this triangle all the time with emerging and ongoing diseases, as it acknowledges that while infectious diseases themselves are caused by microbial agents, the host and environmental interactions contribute to emergence and spread. This triangle actually is also how we think about stopping a disease, as you try to break at least one side of the triangle. You can see all these factors listed under each vertex, and you can essentially map these onto the list of factors that lead to the emergence of a new infectious disease. You'll note that all of these here, if you think about them, all have to do with the interactions between microbes, humans, and the environment. So for example, if we were to look at urbanization, expansion, and population growth, we're talking about, people moving more into the natural environment, leading to more contact with animals and with one another. I do have a lot of slides, so I'm not going to get too much into this particular one, but I did want to mention that there's a new theory out that says that zoonotic spillovers perhaps aren't as rare as we previously thought, and maybe they're happening all the time. There is this great article listed below that makes that case. It's fascinating, and if it's true, it'll change how researchers look for new potential pandemic viruses. Right now, what we do is we look in animals and catalog all the viruses there, 1 million plus, and then try to identify which of those has the potential to jump into humans and then potentially spread from there. But maybe we should just skip that part, and maybe we should just jump straight to humans and see what animal viruses have already jumped over and just perhaps haven't yet evolved the potential to spread. And one point of clarification, not all spillovers lead to pandemics. Most spillovers, if they would spill over into humans, look like the cold or the flu or a GI bug or nothing. And then most of the spilled pathogens then, once they're in, can't adapt to have human-to-human transmission. And then those that do adapt that, most of them can't do it well. If they can, that's how we get a global problem like SARS-CoV-2. Okay, so what are some of the diseases that have recently emerged here in our own state? This list is just a sampling. We also have other things I didn't list on here, like there's multi-drug resistant TB that we've had here. We've had pre-XDR, extensively drug resistant TB here. We've had early signs of resistance and our gonorrhea here. But for the purposes of today, I'm just going to touch on 3 of them. It's going to be hantavirus pulmonary syndrome, Candida auris, and COVID-19. So 1 transmitted through vector, 1 transmitted through contact with surfaces, and 1 transmitted through respiratory droplets and particles. Let's start with the hantavirus pulmonary syndrome. The story here is that in May 1993, an outbreak of an unexplained pulmonary illness occurred in the Four Corner region of the US when a young, healthy, fit Navajo man with shortness of breath was rushed to a New Mexico hospital and he died there rapidly. IHS medical personnel discovered that the young man's fiance had died a few days before with similar symptoms. After a search, more cases were then identified of young, healthy people who had all died after acute respiratory failure. And their tissue samples were sent to the CDC, where virologists linked the pulmonary syndrome with a previously unknown type of hantavirus. And then since all known hantaviruses at that point were transmitted to people by rodents, they figured the vector had to be a rodent. So trapping ensued, and the deer mouse was found to be the main host. And about 30% of the deer mice tested showed evidence of infection with this virus. So by November 1993, that same year, so within months, the specific hantavirus that caused the Four Corners outbreak had been isolated. And in reflection, an IHS physician said, if it hadn't been for that initial pair of people that became sick within a week of each other, we would have never discovered this virus at all. So just pointing out, it was the medical providers that kind of first started pulling this together. This is the map of where cases were found in that first outbreak and a few years after. They think that the sudden cluster that occurred in 93 was climate related. There had been a drought, and then in early 93, there was heavy snow and rainfall, and it helped the plants and animals to just explode. The CDC reports that there were 10 times as many mice in May 93 as there was in May 1992. And so then with that, it was more likely that mice and humans would come in contact with each other, and thus more likely that hantavirus would be transmitted to humans. This is the map now extending to 2006. Thanks for providing that, Dr. Engelthaler. And so now, after emerging here in Arizona and in the Four Corners region, hantavirus is part of our standard public health surveillance and work. It's a reportable condition. We do confirmatory testing at the state lab because the commercial testing can give false positive IgMs. The local health department does case investigations, environmental assessments, and education. And now we have time to reflect a little bit about how new do we think this hantavirus really was? Because it's interesting. While medical professionals weren't familiar with anything like the hantavirus pulmonary syndrome, the Navajo Indians, a number of which contracted hantavirus pulmonary syndrome during that 1993 outbreak, they recognize a similar disease in their medical traditions and actually associate its occurrence with mice. Further, researchers have found evidence of hantavirus in stored lung tissue from people who died previously from unexplained lung disease. And the earliest case so far was found to be in a 38-year-old Utah man in 1959. So perhaps this wasn't the first, emergence of hanta after all. Okay, I'm going to take a break here because I think I should have been taking breaks maybe between the slides, but please, Dr. Engelthaler, you actually worked this outbreak. Any additions from your time and your work with an unknown pathogen?
Thank you, Dr. Villarroel. This is a lot of fun, and hearing this, reminding myself of this history, and thinking about emerging infections. It's really important. Hantavirus was the poster child for the idea of emerging infectious diseases. It really came about right about this time in the 90s because it was this brand new disease that no one had known about, and by a virus that we didn't think existed in the Western Hemisphere. But when we look an evolutionary history of this virus, we actually know that this is a very, very old virus and has likely co-evolved with its specific rodent hosts over the eons. So likely has been in the Western Hemisphere for millions of years alongside with the rodents, and certainly not causing human infections until humans were here, but most likely over the years, over the past several thousand years of humans being in the Western Hemisphere, there were likely outbreaks that would have gone unnoticed because no one was looking for it, but starts to incorporate it into at least the oral history of populations. And so this is a virus in a lot of our diseases that is essentially emerged in our consciousness in 1993, but did not emerge as a disease and had likely been a virus-causing disease for some time.
Great, that was perfect. Thank you. Please jump in after my next one too, which is going to be Candida auris. So Candida species commonly cause bloodstream infections in hospitalized patients and cause significant mortality in ICU patients. Usually it's Candida albicans, but more recently we've been seeing Candida auris, which is a multi-drug resistant species that exhibits resistance to fluconazole and other major antifungals. And the story here starts in 2009 when C. auris was recovered from the ear canals of patients with chronic otitis media in South Korea, hence the term auris. And their report was then followed by them having 2 fatal outcomes of other patients that had C. auris bloodstream infections. Alarmingly, from then, it only took 7 years for C. auris to spread across several countries, including the US. C. auris is concerning to everyone because, one, it seems to have resistance to many or all major antifungals. 2, it can have up to a 40% associated mortality rate. 3, it displays this inter and intra-hospital transmission that is difficult to get in front of. It's now known that C. auris spreads in healthcare settings through contact with contaminated surfaces or equipment, or from person-to-person contact. And actually, the first 7 cases in the United States help to confirm that. C. auris was found in our patients' bodies weeks to months after their initial infection, and it was isolated from their hospital beds, tables, chairs, and windowsills. This is from a CDC report looking at the clinical cases of C. auris rising across the country. You can see years, right, as you move from map to map. As for Arizona's experience, you can see we don't really appear on the map until 2020. So in 2020, Arizona had its first clinical case of C. auris. In 2021, Arizona had its second clinical case of C. auris. And it was found that both of those cases, the one in 2020 and the one in 2021, were both associated with healthcare exposures in other states. Then in 2022, which is not shown on this map, Arizona saw its first C. auris case identified without any healthcare exposures in other states or countries. And by the time 2022 came to a close, there were 205 cases of C. auris reported here in our state. So here's where they're, you can see 2020 is here on the left, 2021, and then the rest of the chart is 2022. Here is where they were identified in our state in 2022. And kind of at this point, we're also wondering the same question as Hanta, did this pathogen exist before and we just hadn't identified it yet? Maybe. But so far, several centers have reviewed archive samples, and there's no sign yet of C. auris. So we don't know that yet. We also don't know why it's independently, almost simultaneously emerging in so many places worldwide. Misuse of antifungals is a possibility, but it is not the smoking gun yet. As is typical for an emerging infectious disease, it has not always been the easiest to identify. C. auris can be misidentified when using traditional phenotypic methods for yeast identification. And so what public health does is we respond to every single case We do confirmatory testing at the state lab, and then we collaborate with the healthcare facility to identify, contain, and prevent future cases. And it's through the screening of exposed persons that we end up identifying colonized patients who themselves can spread it. Clearly, this disease is still emerging here. This is a press release from CDC this past March, and a further spread of CRS in healthcare settings on a worldwide scale is expected. Okay, I'm going to move on to talk about COVID, because we can't go, have this talk without talking about SARS-CoV-2. Right, a respiratory virus that had never before infected humans in a sustained manner, that together with this extraordinary efficiency in transmitting from person to person, and its level of morbidity and mortality, especially among seniors and those with underlying comorbidities, this is the perfect storm, right? Created the perfect storm of an emerging infectious disease. Now, I didn't layer these Arizona case counts against the national curves, but maybe you remember in 2020 when Arizona had early cases compared to the rest of the country, but they weren't followed with an early spread or early surge. And we don't know why. It could be that those early cases we had weren't infectious. It could be that we were just coming off of a fairly severe respiratory season in 2019, 2020, and that somehow was interfering with COVID taking hold. What we do know, though, is once the state opened up, that COVID spread briskly. Arizona was named the international hotspot for COVID-19 in July 2020, and then again in January 2021. During those times, it was like 1 in 100 or 1 in 120 people tested positive in Arizona the previous week. And we don't have this map made just for Arizona, but you can focus on Arizona if you want in these maps. They show the average cases per 100,000 people during the pandemic. They do have this animated on the website noted, but it's kind of upsetting to watch, so I will skip it during your lunch break. But looking at these maps, right, COVID clearly has emerged, and now it's continually evolving and circulating like the flu. And in public health, it's become part of our regular surveillance, like the flu. Okay, I think, Dr. Engelthaler, I'm going to pass it to you to talk a little bit more about COVID variants.
Great, thank you. That's a lot of interesting stories can be told on these emergences, and COVID is no different. Of course, we've been talking about a lot of these stories and understanding the pathogen, the virus itself, the epidemiology of the disease, the changing clinical landscape of this disease. And a lot of that is this truly is, I mean, there's still a lot of discussion about exactly how this virus emerged in human populations, what was the origin of it. It is still a very new virus in human populations. And that means that the virus had to evolve to be able to become adapted and survive in a human population. And humans also have adapted and on a very micro scale evolving to be able to respond to this virus, which seems to be omnipresent at this point around the globe. And so we've been seeing the shifting changes of this virus in what we call variants. And as Dr. Villarroel mentioned at the beginning, that we did have some cases early on in Arizona that didn't go on to spread. And that happened in a few places, especially in the Western states that were coming from China or people who had traveled to China. That very early version did not spread wickedly like the versions we see now, Omicron. And so it took a little bit of changing and evolution. And we know that, some early mutations caused this thing to explode in Europe. And then the versions that came out of Europe really have been the dominant version. And then there's been newer variants of that. And we talk about alpha and delta and beta and gamma. You remember these throughout the pandemic. Well, we're in Omicron land now. That's the last major variant, and everything we've seen since January 2022 is Omicron. But there's different subvariants as the virus continues to change. And so I want to go ahead and click next. I just want to talk a little bit about this. We've tracked these variants all along. The newest one, if anybody's watching this probably too closely, they've seen that CDC or WHO just named a new variant of interest, which is this BA 2.86 or JN1. And that's just because it is becoming dominant in places it shows up. Certainly in Europe is becoming dominant. In the US, the CDC estimates about 8-9% of the circulating variants right now are this 2.86. And then in Arizona, we've actually seen it here, but only in a half a dozen cases. So, just under 3% of the total viruses that have been sequenced in, over the past month and a half. So this is essentially just looking at the shifting landscape. We look at the chart, that's the COVID sequencing dashboard that has been going really since the beginning of the pandemic. We can see all those different colors. Those are just different flavors of Omicron. This new version is still Omicron. It does have an interesting spike mutation that we really haven't seen previously, but I'll talk about spike mutations here in a second. But it is still acting exactly like Omicron, very high transmission and relatively speaking, low severity for almost everybody, probably for a couple of reasons. One, because almost everybody has some level of immunity, circulating antibody. They don't work great in a lot of cases, but your cellular immunity prevents it from being severe. But also, this virus, since it's become Omicron, is not a lower respiratory disease in almost every case. There are obviously outliers. The vast majority of cases are upper respiratory infection and transmission, which allows it to move in and transmit out really quickly. It prefers upper airway epithelial cells as opposed to lower airway epithelial cells. So this virus did change when it became Omicron. It became a very different virus in the disease that it causes, and the epidemiology of it, how it spreads to pretty much everybody everywhere. So click on the next one, please. And I'm going to be sorry. Dr. Villarroel is going to be driving, so I'll be saying click or forward a lot. So when we think about these variants, I'm starting to consider these as just mutation vehicles. So these variants have these mutations that show up that give them some type of selective advantage. And now that's typically to escape antibody because it's already has the best advantage possible as far as how it infects cells and spreads and replicates. We haven't seen anything faster, and it just stays in the upper respiratory airway for the most part. And so it doesn't need to change there, but it does need to adapt to antibodies. So those places where the antibodies bind, and you're looking at the spike protein there in the middle, on the top of that, that's the receptor binding domain, where it would really essentially connect to human cell receptors and then invade cells. But that's also where antibodies will glom onto to prevent that infection from happening, that cellular infection. And so if it could just change enough that the antibodies don't attach very well, but still gets into the cells, that's what it's doing. So there's just some selected mutations that seem to keep reoccurring. We see these showing up in new variants, the old mutations that are being reselected for. So the mutations are recurring, but the variants are becoming extinct. We don't see, when it becomes extinct, we don't see that variant again. It just keeps evolving, but it has some nice mutations that it's using over and over again. And so that's some new research that we're working on. Next slide. And then I want to point out too that testing is way down. Of course, we're not doing really controlled testing. A lot of at-home testing. We don't really have good ideas on how much COVID is circulating, except really exciting wastewater surveillance, which is now occurring in many parts around the globe, but certainly throughout the US, including here in Arizona, and has been really since the beginning of the pandemic in some cases. And we can actually see how much COVID is in a population by looking at wastewater. And so here on the top row you see on the right-hand side, that is really the most recent months. The gold or orange color is Arizona numbers, which seems to be dropping a little bit, which matches the purple, which is really kind of the Southwest region. The black line is the US. So it looks like it's increasing overall in the US, which also isn't too surprising, but not drastically. But it is going to be a good measure for us moving forward of what's really happening in our population. And really exciting is that this is being done not just for COVID, but for many viruses, and in fact, other pathogens too. We are seeing this as going to be one of the great surveillance tools for public health moving forward. That bottom is another dashboard. Dr. Crystal Hepp's team has been monitoring Flagstaff and some other municipalities for multiple viruses, and that could be checked out too. We have the links as well. Next slide. So I'm going to move into now the next topic that was listed before, which is the current emerging infections or the future emerging infections in Arizona. So we're going to try to be a little bit predictive without trying to be too scary. So I want to touch on dengue, which we know is actually emerging, and now a new Southwest cryptococcosis. So Cryptococcus gattii, we'll talk about. Arizona has its own special brand of that we are just finding out about. High-path avian influenza, and we'll touch on chronic wasting disease as well. So next slide. So dengue is a flavivirus that we typically think of as being in Latin America or in Africa in some places. But this is spread by a very specific mosquito or a couple of mosquitoes, and that's Aedes aegypti. So we can't have dengue virus, unless we have, one, the virus in humans, and two, the vector that can transmit it by picking up from one human and giving it to another. This isn't spread in birds like West Nile virus, so it doesn't just maintain itself in the environment. But we have to have the mosquito as well as the virus. So first step was the mosquito, and it reemerged, I should say, in Arizona in the 1990s. Then we reported on, and we think that that's somewhat strange. You know, this is a very tropical type of mosquito. We look at Arizona, the Sonoran Desert landscape is not good for Aedes aegypti. But when we make it like that picture on the right-hand side there, that's what Phoenix looks like now, with lots of ornamentals, lots of imported plant life and humans living in a lot of water sources. It's now become a great source for mosquitoes, and probably the number one source for West Nile in the country. But now what about Aedes aegypti? So we've seen it here, first detected after 50 years in the 90s. Next. We can see on this map here, this is Maricopa County, 2012 through 2021, the spreading prevalence of Aedes aegypti showing up in traps run by the Maricopa County vector control. So this is a mosquito that is just blossoming and now become and is an endemic mosquito in Maricopa County. All we needed was to have the virus now present in individuals. And as we have people move up and through either travel or migration from Mexico and further south, they can bring the virus with them and then have the mosquito transmit. And lo and behold, click on the next, please. We actually had this show up, a locally acquired dengue virus in Maricopa County last year for the first time ever known in Maricopa County. So for Arizona, and that's probably not just a one-off, because we do have more and more individuals that do have dengue virus. We were able to detect those. They're travelers or migrants, but they're going to get exposed to mosquitoes, and we're going to see more transmission. So during that investigation, they found evidence of several people in that same neighborhood also had been exposed to dengue virus. Next. And also California this year had its very first cases of dengue virus twice, once in Pasadena, once in Long Beach. So they're starting to see local spread of dengue, which is somewhat predictable. Next, because we do have a fair amount of dengue south of us. And in fact, Mexico's dengue hotspot is in Sonora, Mexico, which is where we obviously have the most movement of populations from Arizona, are moving into Arizona or traveling, and therefore can be bringing the virus with and causing more and more transmission. So we're just going to have to be on the lookout for this. It will continue to grow in my estimation and become an emerged infection in Arizona. Next slide. Okay, so want to talk about another fungal infection. The Candida auris is a fascinating emerging infectious disease, and by everything that could be seen, it is a new fungus that is rapidly spread around the world, and it changed as well too. As we have seen with some of these viruses, or excuse me, pathogens, they can evolve, and fungi typically evolve slower. But what's really interesting is, as was mentioned, the first cases we're seeing in Korea and Japan, and that was an ear infection. Well, Candida typically is an environmental pathogen and doesn't like the warm body temperatures, and wouldn't normally cause a systemic infection. Well, it seems to have now been adapted and able to cause systemic infections in humans in a way that we wouldn't have expected. So it does look like it has changed. We have seen that fungus in the environment, and now recently reported on detected in the ears of dogs in India and probably elsewhere. So it is probably something that's been in the environment. We didn't detect it, but just looking at the evolution of it, we know that it has changed and emerged to become a human pathogen. Cryptococcus is typically, we think of for infectious disease, Cryptococcus gattii and Cryptococcus neoformans. And I'll talk about that in a second. But in Arizona, we look like we may even have a new species of Cryptococcus that is causing human infections. So let's dive into that a little bit. Next slide. So Cryptococcus gattii is an environmental fungus that typically is found in decaying plant matter, in tree holes, or decaying tree and wood. And so it has really been seen as a tropical or subtropical pathogen, or excuse me, fungus that just fine right in the environment. It doesn't need a host species, but it can infect mammals and cause severe disease. Usually when you have spores or desiccated yeast gets stirred up into the air from that decaying plant matter, breathed in, becomes a pulmonary infection, and then can fairly easily get into the bloodstream, travel to other areas, but most importantly into the CSF, and then cause meningitis or encephalitis. Cryptococcus is not been thought to be, Cryptococcus gattii, not thought to be a really important environmental fungus in North America, except about 20 years ago, we saw a couple of new versions of this show up in the Pacific Northwest. We did a lot of studies on that, very interesting. I'm not going to discuss that. That's a whole different interesting story. But Washington, Oregon, Vancouver, and then some areas in coastal California with another subtype of Cryptococcus, but certainly not in the desert. So it doesn't make sense, usually in a more wet, moist environment. Next slide. Okay, so this looks real. This is an eye chart, but really what it is, it's a family tree. We use phylogenetics a lot when we use genomics to really understand these organisms, how the different species, how they're related or different from each other. The two main ones I mentioned, Cryptococcus gattii and neoformans, those are really complexes of species now that we're learning as we dive further into this. And then they each had a number of subtypes that we knew about. These were labeled as, in gattii cases, VG1 or VG2, just to separate them out. We knew that they were significantly different. They've now been named as separate species. And you can see that circled one there was just one strain that was available that was so different than everything else. It was named by a group in the Netherlands as a separate species, but it was related to what we were calling VG3. And so that'll be important in a second. Go ahead and click next. So when we did some analysis on this, we saw that VG3, and there were we've identified a couple of strains that seemed identical to each other, and they came off that VG3 branch, but still very different. and didn't really understand the importance of that. Next, you see over on the left-hand side, where that blue arrow is at the top, a group of colleagues in the UK had actually identified a new subtype, or actually species. It was called VG5, but found with hyraxes only in one place in sub-Saharan Africa, and really only found not in decaying trees, but in their middens and associated with their guano. Cryptococcus neoformans associated with pigeon guano, interestingly enough, but not typically the other Cryptococcus gattii. Well, this one seems to be associated with hyrax guano in one location in Africa. But during that same study, they had also included that group that you can see in the red. Click one more, I think. Yeah, that is circled there. That VG6, what they called VG6 for the first time, and somebody else had called the Cryptococcus decagattii. Sorry about all the nomenclature. It's really only interesting to the Cryptococcus people, but these are new distinct species and we think could be different. We didn't know anything about this. There seemed to be one strain that had been replicated a couple of times in the lab. Nobody knew really much about it, other than that strain originated from Mexico. Next slide. So now we talk about Arizona. Let's go into the way back a little bit. 2011, a Tucson veterinarian identified Cryptococcus in a cat, and then it was later confirmed to be Cryptococcus gattii by a colleague of mine in California, but it was just sitting on a shelf. Gattii, nothing really new about it. Didn't really know. Maybe it got gattii from someplace else, the Pacific Northwest or something, and caused an infection in a cat. Well, fast forward 2019. There were 2 cases of Cryptococcus gattii that were in immunocompromised patients that did not seem to have any travel. In fact, both of these, I think, were from the southern part of Arizona. One of them was pulmonary. One was a pulmonary case. One was a meningitis case. I think that was a fatality. And then a third human case of gattii showed up in 2021, also causing meningitis. Also an immune-compromised individual. When we were able to get those strains from those patients, we did the sequencing and showed that this is part of that VG6 group, that Cryptococcus decagattii, that there was only that one strain, seemingly from Mexico. So we think we have something new on our hands. We got the cat case. We tested that too, and shows up. Go ahead and click, I think, on the next one. In the top there, where you see in the red, it says VG6. That is that new group, this decagattii showing up in Arizona. Go ahead and click. This is just a blow up of that grouping. And so we actually are starting to better understand this. We learned that the Mexico cases, there's actually 3 Mexico cases that are in this group, almost all identical to each other, but 3 different individuals, as well as the Arizona cases, and then a very strange sample from 2017 from Argentina. We don't understand that yet, but clearly a southwestern Arizona, perhaps Arizona, Sonora, Mexico, we don't know where in Mexico, Cryptococcus that is causing disease in cats and humans. Next slide. And so what do we know about the epidemiology and ecology? There's no good epi data yet on these individuals. That it's been a little difficult to get that information or travel history, other than we think the very limited possible travel. The veterinary cases, we've seen now all of those down in the Tucson region in neighborhoods that look like this, where it's, pretty much their average suburban neighborhood in Tucson. So we have some working hypothesis that it could be a local rodent or small mammal reservoir, wood rat nests, just like the hyrax middens. That could be a potential good place for this fungus to survive and thrive. Maybe in decaying local flora that you have decaying saguaros and some trees. It's not as prevalent as you would see in highly forested areas like in the tropics, but that's possible. Could be another vertebrate reservoir vector that we don't know of that's naturally in the environment, or some other microclimate habitat in the desert Southwest that we just haven't looked at. But currently we still know very little. Next slide. So we've actually instituted some surveillance with veterinarians to look for more cats, because cats are going to be a great sentinel species for us to to be able to maybe nail down the epidemiology, because they don't move around very much. And then when they do, we typically know where they've moved. And so we've reached out and have worked with veterinarians in the Tucson region. We've identified 3 more cats that have had Cryptococcus gaddii, 2 of them typed to be this VG6. So we know that we're going to continue to see this, and the more we look, the more we're going to get. So there's still more to learn about this. Next slide. But for now, I think we really need to have a clinical suspicion of gaddii. When you have a Cryptococcus infection, still seems to show up like other cryptococcal infections, either pulmonary or neurologic disease. Any suggestion of that, please contact public health, report it to local and state public health. Try to get that travel history, any other relevant epi information. And then let's get some samples, because we do need the sequencing to confirm that it is part of this new species that we're seeing in Arizona. And we're working with ADHS to do that. Next slide. Okay. Sorry, that was probably longer on Cryptococcus than I planned, but it's fascinating, and it's Arizona's new emerging disease. Now let's talk about something more global, highly pathogenic avian influenza, which is a panzootic. So next slide. We all know that there are different types of influenzas, the big groupings, influenza A, B. There's also C and D. We don't hear very much about those, but what we normally see transmitted in humans is A and B. These are all influenza viruses are most likely originating out of birds. This is a bird virus, but some have moved into other mammals, including humans, and then adapted in that environment to be able to spread between them and become, endemic infections like our H1N1 or H3N2. I'm going to say H and N a lot, as we know those are the glycoproteins on the outside of the virus that your antibodies would connect with, but also how it actually interacts with human cells, just like we talked about the spike protein with COVID. Those are really important as they change. Not only they can change whole different versions of these, so you get an H1 or an H5, but you also have mutations, what we call mutational drift occurring within those different genes that actually can make it more or less infectious, or be able to evade antibodies that we had the previous year, just like with COVID. But importantly, with avian influenza, we're talking about H5N1, which is a bird virus that we know is highly fatal in humans. We've seen outbreaks in the past, but not outbreaks between humans. It's just when humans come in contact with birds. And that's what's kind of starting to happen more and more now. So let's talk about high path avian flu. Next slide. So this H5N1 high path avian influenza has been spreading in waves of the past couple of decades. But the most recent wave that's been occurring started in 2020, and that's all the data you see here. In the upper right-hand corner, that's the global picture of where this is really shown up. Everything in red is H5N1 spread around the globe in the last couple of years, last 3 years, I should say. The upper left-hand corner, that's in the US. That is where we've seen it in birds. It has shown up in every state in the US, including Arizona, causing bird infections. But what about mammal infections? That's where we're going to get concerned about a new human emerging disease. Well, the lower right-hand corner, we can see mammal infections have occurred in many places around the globe. And then we look more closely in the US, every place you see a spot, that is showing where it has shown up in mammals. Now, I know you can't read that because it's too small, but on that legend in that lower left-hand slide or image, that legend names the different species. Every one of those species is a carnivore. So that tells us, and this gives us a good hint how mammals are getting it. They're eating infected birds and maybe other infected animals, but definitely infected birds. And then they get it through that kind of severe exposure you get from eating a carcass. It could be through cuts, it could be through ingestion, multiple ways. And then the mammals are seen acting weird, dying, and then tested. And so we can see that many places this has occurred. So we're missing the vast majority of those because that's happening out there in nature. But it has occurred in almost all the states around Arizona. That tells us that it's most likely has occurred in Arizona mammals. We just haven't identified it or seen it yet, but it's a good bet that it's here already. Next slide. And this is just showing the birds in Arizona. So I think that 56 birds had been identified by the last count in Arizona, including birds in zoos, but also backyard poultry and natural birds, wild birds in Arizona, and importantly in the condors. So this is really significant, as you know, and have heard the condor release, how we've really brought this bird back from the very edge of extinction. And then there's been 21 cases of condors who have got it, and the majority of those have been fatal. There have been several that have survived. So there's a lot of vaccination efforts going on to save the condors again, but also vaccines are now being used more and more in bird flocks, as well as culling. This is so significant that there's probably been about half a billion birds, poultry birds, have either died or have been culled since this last wave started. So this is an enormous impact on the economy and on animal life, but also the potential risk for humans. So next slide. Now, there hasn't been very many cases. There's been 17 cases documented since the beginning of 2022 around the world. 8 of those have been serious, 4 have been deaths. So remember, H5N1, when it causes a human infection, it's much more serious than the human flus, but it doesn't transmit easily between humans. Almost all of those have been traced back to close bird contact. There's been no known human to human or mammal to human spread. So that's the good news. There was one case in the US, it was a mild case in Colorado. It is susceptible, good news, susceptible to all influenza antivirals. So we'll be watching that pretty closely. And there's been no human, there is no human antibody protection to H5, but we have a lot of circulating N1 because we have H1N1, and the antibodies that we have from that actually are somewhat protective against this, but not against the H5. Next slide. And then we see that here's the important thing, is that this virus will continue to not just mutate, but it actually mixes with other influenza viruses. This is what influenza does. And the ones that are dominantly in the US, that blue group down at the bottom, actually are now more severe, causing more severe mammal infections than the original strain that we got coming from Eurasia. So this virus is changing, and there is a risk that it could mix with a swine flu virus or a human flu virus and become something that's spread between humans. My last flu slide coming up here. We might just make it more transmissible on purpose. We've been doing this in the lab. Fortunately or unfortunately, this was actually a really big deal that there were some researchers in Europe as well as in Japan back in about 2012 that were trying to figure out what does it take to make H5N1 transmissible between mammals using ferret model, which is a close good model for human respiratory transmission, and they were able to do it. Thankfully, no lab escapes, no lab leaks. We did not get a giant pandemic, but it was potential that we could. Can you click one? And really interesting, just last month or a little over a month ago, a report came out that it has gotten into minks, which is essentially the very close cousins of ferrets, and spread in minks. So there's seemed to be mammal to mammal transmission able to occur, and you could actually get some adaptation in situations like this. So we can't think that this is just a bird problem and there's just one-offs. We have to be watching this very closely. Our next slide. And then I'm going to do real quick, chronic wasting disease, not an emerging zoonotic disease because we're not getting it, but it's really important to know about. Next slide. This is a infectious protein, not a microbe, but an infectious protein. These are prion proteins that all mammals have, and you have normal prion proteins. But if it touches an infected or a diseased protein, or what we call a diseased prion, that diseased prion can change the shape of the good one, and it starts to break down cells in the brain, and you start to get this spongiform effect. And we've seen this time and time again. Mad cow disease is the most famous one, where that was actually a sheep version of this that actually got into cattle, the way they were handling cattle, feeding it sheep offal. And then it got into the cattle supply and caused a big outbreak in the UK back in the 1990s. That's been stopped, but you can get it from ingestion. Well, we're seeing a prion disease, can you click on the next one, showing up in deer and elk in North America, and has been spreading. We have not seen it yet in Arizona, but again, it's in the other four corner states it's most likely going to be found in Arizona. And the biggest risk is going to be the handling of these animals, primarily by hunters, and then potentially ingestion. And so there is a potential for a zoonotic risk. I think that one's still low because we haven't seen any human cases yet anywhere, but it is potential. So we got to be watching it. I will turn it back over to you, Dr. Villarroel.
That's great. All right, we're going to fly through just the very last part of these. And real quick, before we get into the resources, I thought we should touch briefly on the detection systems we have here in Arizona and how we could help, detect these emerging diseases. Generally, we could classify these into laboratory and human detection systems that I was going to talk about. Let's skip laboratory for now. There's animal surveillance here as well that I'm not highlighting, and let's instead just talk a little bit about human surveillance. So there is a list of reportable diseases that providers report to public health. And note that all these conditions, right, can be either confirmed or just suspected. I will point out the glorious catch-all in this list, which is emerging and exotic diseases, and it has a phone icon. So if clinicians suspect something new occurring, they should call public health immediately. We also have school and childcare and shelter reportable conditions, which include some potentially re-emerging diseases. We have biosense data where we can pick up the real-time trends and the symptoms of those seeking care in EDs, urgent care, and hospitals. But really, all this, most of which we didn't talk about, but all of these systems of detection really work best with some kind of spark or a direction or a clue. It works best when there's an identified situation in another country or in another state, or if a clinician calls to alert us to something. So the astute clinician, this is a term that they now have an award for, and it refers to a clinician who notices and reports on an unusual occurrence. The IHS physician talked about that, right, with hantavirus. And we do see it all the time here. Most recently for me personally was during 2021 when a nurse called in to say that her ICU was being taken over by West Nile patients, which was not the area where we were necessarily seeing the vector distribution. And it had us looking a little bit more closely. To be honest, the clinician call is very often our catalyst to detecting a new disease or a significant outbreak. Touching briefly here on the resources, this is the list of urgent resources. So if you have a question, a suspicion, anything around an emerging infectious disease, please call public health. I put the 4 largest health department numbers there. You can just plug it into your phone. That's what I have. That's what I've done. And then if there's a situation in Arizona, we will send out a health alert to registered clinicians. There's the website. The alerts go out super rarely and are specifically catered to physicians here in Arizona based on what we are seeing here locally. I think the last one was on congenital syphilis. And then there's a number of websites you can check on your own time on the left. The dashboards in yellow are the things that Dr. Engelthaler and I get asked the most about. And then on the right, please feel free to join any of these conferences. The top one, ARIDS, is always a winner. It's with the ID physicians, and they go through case studies and the newest literature. Coxie Study Group is unique to the Southwest. AZID is also a favorite of ours. It's all infectious diseases with a public health lens. And so we have time just for our last 2 take-home messages. From me, the summary for me is that emerging, re-emerging, whatever, all these diseases continue to place a substantial burden on health globally and here in Arizona. My take-home to clinicians on the call is super practical, and it's just to please keep caring for Arizona residents and their hypertension, diabetes, substance use, asthma, anxiety, depression, and loneliness. It's not important that you know anything specific from today, in my opinion, other than who to call when you need to. Because for public health, clinicians are the most effective sentinel surveillance that exists. The automatic lab reporting cannot do what you do. AI cannot do what you do. Only you can call to alert us to something that does not fit a pattern. It's how we found Hanta. It's how we actually found COVID-19 in our state as well. For fun, I noted some extra reading. Phase 6 is a novel. It's fabulous about emerging disease. Spillover is academic, but it is readable. The Great Influenza book, it's on every single public health reading list, but it is a beast. And I linked my favorite NPR article here as well. Okay, Dave, close us out.
Thank you, Dr. Villarroel. That was great. And I love those books as well. So my take homes, 2 quickly ones. new pathogens and their diseases will continue to emerge and reemerge. We can't just think that we're one and done with some of these. They do reemerge. We got to continue to look out for the new while we're taking care of the old. And we don't need to help them along, which we do in many ways, either by, not as good biosafety in the lab, but also, going out and taking over new places in nature, exposing ourselves to new viruses and bringing those back into the population. Given that we need faster and smarter tools to gain intel on these pathogens, to allow us to respond both faster and smarter. So while absolutely we love and need the astute clinician, they're critical, we also need to bring in better tools to support them, to help them identify patterns and data that's going to be useful to respond quicker so that we can save more lives in the future.
Dr. Villarroel, Dr. Engelthaler, thank you so much. We could have spent an additional hour or more on that, or days. We are so appreciative of that. We want to thank all of our participants for joining us today. And we will be sending out some information following this, where you can link to NARBA's website for the slides, today's recording, and also information about CEUs. So thank you all very much. Happy holidays, and we'll get back to you in 2024 with next year's lineup of Arizona Community Grand Rounds. Thank you all so much. Have a great day.